Beyond group selection. The concept of the generative phenotype calls for a broader appreciation of the object of selection. In the past, passionate debates among proponents of the modern synthesis concerning the validity of the group selection have persisted for decades. Today, there is widespread acceptance of the concept. In the US, we need to go beyond the concept of group selection because EEP calls for a broader understanding of the nuanced ways in which selection acts over many generations. Also, among prokaryotes, the concept of the individual genome, the individual gene pool, and even the concept of a species have been supplanted by widespread acceptance of horizontal gene transfer and group phenomena such as quorum sensing and biofilms. Since genetic information is regularly shared among multiple species, and since individual fitness often depends on group interactions and group behaviors, the question of whether natural selection acts at the level of individuals, populations, or species is rendered moot. What about among multicellular eukaryotes? In Chapter 2, the transcendent quality of emergent evolutionary potential, EEP, was discussed. EEP is not limited to actual events that take place in time and space, but also includes constantly changing logical relationships that transcend space and time. The generative phenotype is not only subject to the consequences of EEP, but provides illustrative and perhaps quintessential examples of it. Developmental mechanisms in the genomic scrapyard represent toolkits that are reused in various ways in various lineages. The flexibility, robustness, and efficacy of developmental mechanisms and their genetic determinants, and their ability to be reused in different ways, can make the difference between long-term survival of highly branched lineages over many millions of years and rapid extinction. Extinction is a normal aspect of evolution, and extinct species greatly outnumber extant ones. Part of the reason for this is that the generative phenotype affects evolvability. Evolvability represents an aspect of natural selection that transcends individuals, populations, and species, but cannot always respond quickly enough to rapid environmental changes, such as those caused by human activities. Global warming is, for example, a significant factor in the recent bleaching of coral reefs on a global scale. Co-option and exaptation are just two of the widely recognized aspects of deep homology that are now central concepts in evolutionary developmental biology. The generative phenotype represents EEP that is certainly subject to natural selection, but is more transcendent than the ecological phenotype. Why distinguish between the ecological phenotype and the generative phenotype? The ecological phenotype refers primarily to the fitness of the phenotypic gestalt of the developed individual. The efficacy of the structures and functions of the individual within a particular niche affects its survival and reproduction. This usually involves actual environmental events that take place following the embryonic development of the individual. The generative phenotype, in contrast, refers primarily to the ways that toolkit elements are deployed in particular individuals to produce that gestalt. This distinction helps to clarify the relationship between evolution, development, and the phenotype of the individual. Both concepts help us to understand both the short-term, real-time events, as well as longer timeframes responsible for evolution. These various timeframes have long been recognized by evolutionary theorists in the context of the modern synthesis. But prior to our present-day understanding of the molecular, cellular, and developmental basis of the generative phenotype, the longer time frame of natural selection has been enigmatic. The Updated Evolutionary Synthesis, UES, provides a deeper understanding, primarily thanks to the new and active field of evolutionary developmental biology, EVO-DEVO. This is elaborated further in Chapter 15. The GA demonstrates the evolutionary significance and origins of the phenotypic gestalt. As discussed in Chapter 13, the genetic algorithm, GA, is a software algorithm that emulates natural selection because selection is blind and path-independent. It does not depend on the way that the combinatorial parameters are recombined. Similarly, biological selection acts on the final combinatorial product, the phenotypic gestalt, the product of numerous interacting elements of the generative phenotype. Thanks to SSM-IBE, the generative elements of the biological phenotype have extraordinary organizing power. Multiple variables contribute to the gestalt that is the target of selection. The multiplicity of variables is especially pronounced when natural selection acts on determinants that influence development, simply because the molecular, cellular, and developmental toolkits, that is, the numerous contributors to the generative phenotype, are complex, redundant, and robust. In virtually every case involving the ecological phenotype, there are numerous overlapping, synergistic, cooperative, and combined molecular, cellular, and developmental interactions that ultimately contribute to the ecological phenotype. Given the sheer complexity and diversity of elements that contribute to the ecological phenotype, and that are part of the generative phenotype, it is truly remarkable that we are able to understand as much as we do about the detailed, concrete mechanisms that are involved. That is because biologists have, over many decades, developed extremely powerful empirical techniques for isolating, tracking, and detecting specific interacting components. This is routinely accomplished in literally thousands of biology laboratories throughout the world, on a daily basis. One of Albert Einstein's most famous quotations, and he had many, is often rephrased as follows: "The most incomprehensible thing about the universe is that it is comprehensible." Our ability to understand the universe is astounding. It's important to acknowledge, however, that this comprehensibility has a historical component. In the biology lab, the reason that it is comprehensible is a triumph of the scientific method, of naturalism, and the professionalism of the thousands of unsung scientists who have developed and tirelessly applied powerful empirical techniques, careful judgment, logic, and reason to their experiments and observations, both in the laboratory and in the field. The processes of fertilization, cleavage, gastrulation, and organogenesis are all high-level cellular descriptions of general stages in the reproduction of complexity by the generative phenotype. Cytoplasmic determinants, morphogens, and transcription factors are all involved in molecular interactions required for complex organization to arise in the multicellular embryo. Refer to Chapter 10. For Rethinking Evolution and for the Updated Evolutionary Synthesis, UES, the problem is not that there is not enough detailed empirical information about developmental mechanisms and the evolution of those mechanisms. The problem is that there is far too much detailed information to do it justice. In Chapter 15, we will explore the powerful synergy between developmental and evolutionary biology.